Hi, my name is Chandramouli. I am an assistant professor of psychological and brain sciences and anatomy and neurobiology in the medical school. I run a lab which studies decision-making in primate models. So I run a monkey lab, and typically I'm interested in understanding the process of perceptual decisions. Perceptual decisions is something we do every day in our life. For example, if you cross the street in the Commonwealth app, you're often looking to your left or your right to see what cars are coming, and you choose the appropriate actions in order to achieve your behavioral goal of crossing the street. So I'm interested in how different brain areas in the monkey brain actually support this particular process. And in particular, we're interested in these brain areas such as the dorsolateral prefrontal cortex and the dorsal premotor cortex. These are brain areas which are often impacted in stroke and also in neurodegenerative diseases. One of the things we do is we often train our macaque monkeys to perform tasks which involve perceptual decisions. So for instance, in our particular case, these monkeys are discriminating the dominant color in a central static checkerboard composed of red and green squares. And then more red than green, he touches red target, more green than red, he touches the green target. We randomize the target configurations so that we can decouple what the stimulus is showing to what the actions the animal is actually performing. Right, and that'll become important in a second. Then we put in the best electrodes we can get. Right now, we've gotten neuropixels into our primate brains, which allow us to record from more than 384 channels across like many millimeters of cortex. And so what do we do with all this rich neuroscience data? So we have very interesting, tightly controlled behaviors from these monkeys, and we have lots of neural activity recorded from these brain areas. And we often use machine learning methods to try to understand how these decisions unfold on single trials. I'll leave a lot of the mathematical details, but I would like to show you, this is actually behavior of my monkeys when they responded quickly on some trials in the decision-making task and responded slowly on other trials in the decision-making task. And this is data from the dorsal premotor cortex. And what you'll probably notice is that in the trials with the red trajectories, those are actually slow reaction time trials. So this is actually this is, as Emily mentioned, the latent activity of small populations of neurons. And you can see the separation even in state space, even before the checkerboard comes on. And what causes such fluctuations of the initial state before even the checkerboard comes on is actually what the animal made. If an animal made a mistake on the previous trial, this leads to the slowing down of the behavior of the animal, and it also changes the neural activity in these brain areas. Okay, multiple brain areas actually support decision making. So how do we study how these areas actually contribute to decision making? So if you actually look at brain areas such as the dorsolateral prefrontal cortex and dorsal premotor cortex, you can use methods such as dimensionality reduction, such as principal components analysis, tensor component analysis. The list of methods is growing every day. And in this particular case, these are two different brain areas, DLPFC and PMD. And remember how I told you how I randomized the target configurations? In a motor area, all we see is whether the animal chose to go left or right. But in the prefrontal cortical area, we can often decode what color the animal perceived and what target configurations actually see. Okay. The other thing is I've talked about how populations of neurons actually perform these kinds of tasks, but I'm also interested in the individual cell components which actually perform these tasks, right? So what we've been developing in the lab is a way to take waveforms from these high density electrodes and try to use data science methods, specifically if you're interested in it, in weighted nearest neighbors approaches, to try to identify cell types from waveforms. And this is a paper which was published by my graduate student, Kenji, and we were able to go way beyond the classical approaches of just identifying two candidate cell types and identify many different cell types in these brain areas. Finally, you know how multiple brain areas are actually involved in this process of, I'll read these details, you know how multiple brain areas are involved in perceptual decision-making. How do we build hypotheses about how these areas actually interact? So one of the things my lab does is to build artificial neural networks where we connect artificial neural networks to, like, let's say, dorsolateral prefrontal cortex to dorsal premotor cortex, train them to perform the same tasks as our monkeys. And then now we can reverse engineer these networks to understand what happens. And so this is our model, and the model makes predictions of what the data should look like. And if you remember what the data look like, this is the data from the right-hand side shows you the data from premotor cortex, and the left-hand side shows you data from the dorsolateral prefrontal cortex. If you notice in premotor cortex, there's no real encoding of choice or encoding of color or target configuration. But in contrast, in the dorsolateral prefrontal cortex, we see in the model and the data what the actual similarities are between those two. So my lab really lives at this intersection of data science and neuroscience. We train all of these animals to perform sophisticated tasks, and we get this rich neuroscience data set, and then the game is afoot, basically, to try to link this neural activity in these areas from hundreds of neurons often to like specific tightly controlled behaviors on each trial. So thank you.